What's going on, Science 10s? It's Mr. Walton here, and today we're going to be talking about some microscope calculations. That's right, you're going to have to take out your calculators again. Believe me, this is only the second last time you're going to have to do it. As well as today, we're going to be learning how to prepare a sample to view under a microscope with a technique called preparing a wet mount slide. The first thing we're going to talk about today is magnification. Magnification is the apparent increase in size of an object when looking through a microscope or a telescope. When we look through a microscope, the objects that you observe will appear much larger than they are without the microscope. And we can quantify how much bigger something will appear by describing the magnification of the image. An example of magnification can be seen here. On the left, we see an image of a $5 bill without any magnification. On the right, we can see that it appears approximately 10 times larger than it does on the left. The image on the right appears to be magnified by 10 times, so we say that it has a magnification of 10x. You can see more of the object in the picture on the left, but you can see more detail in the image on the picture on the right. Both images contain useful information. We can predict how much an object will appear to be magnified by taking the magnification of both of our lenses and multiplying them together. For example, when this microscope is on low power, like you see here, the light from the sample passes through a 4x objective lens and a 10x ocular lens. That means our image should appear 4 times 10, 40 times larger than it would with the naked eye. When we change the microscope to a medium power, so we're changing the objective lens, we use a 10x objective, but we use the same 10x ocular lens we did before. So 10 times 10, we should get a magnification of 100x. High power uses 40x objective with the same 10x ocular lens, so we'll get a magnification of 400x. Our data booklet defines the approximate field diameter for microscopes at Lindsay Thurber. They can be found on page 3 of your data booklet. For example, when we look at this image of a flea on low power, the approximate diameter of our field of view is 4,400 micrometers. We use the letter U or the Greek letter mu to represent micro. Micro just means 1/1000th of a milli. So a micrometer is 1/1000th of a millimeter, or 1,000 micrometers is 1 millimeter. When we increase the magnification to be medium power, we can only see 1,720 micrometers, so we can see less of the overall picture. When we increase again the magnification to be high power, we can only see 440 micrometers. As you can see from these 3 magnifications of the same image, we can see less of the total flea each time, but we can see greater detail with each increase in magnification. Knowing our field diameter gives us some perspective of how large the objects in our field of view are. This allows us to estimate the size of these objects by guessing how many of the objects would fit side by side, called the fit number, and dividing the field diameter by the fit number. This is a very subjective thing to do, and it requires you to do a little bit of guessing, but it also gives us a general understanding of the perspective of these objects. The equation for estimated size can also be found on page 3 of your data booklet. For example, this is an image of a salt crystal under medium power magnification. That means our field diameter is approximately 1,720 micrometers. If we want to estimate the size of the salt crystal, we need the fit number of the salt crystal. How many of these salt crystals do you think could fit side by side in our field of view? When I look at the image, it appears to me that we might be able to fit about 4 of them across our field of view. That means we can estimate the size of our salt crystal by taking our field diameter, which is 1,720 micrometers, and dividing it by our fit number, which is 4, we should get an estimated size of about 430 micrometers. That means we've estimated the size of this salt crystal to be about 430 micrometers wide. Take a moment to pause the video and calculate the estimated size of the following paramecium under high power. Our field diameter under high power is 440 micrometers, and it appears to me that we can fit approximately 3 paramecium across the entire field of view. So we take 440 micrometers, divide it by 3 paramecium, and that means we should get approximately 147 micrometers. Given the information we learned yesterday about cell theory, 1, all life is made of cells, 2, cells are the building blocks of life, and 3, all cells are made from pre-existing cells, we would like to take a look at something that is living, or at least used to be living, and we're going to look at it under the microscope. I've chosen an onion because you can very easily remove thin layers of the onion without having to put too much effort into it. The method that we're going to be using to prepare our sample is called preparing a wet mount slide. This means that we're going to be taking our sample and it's going to be surrounded by water and sandwiched between 2 clear layers of glass or plastic. The first step is to get a very thin layer of onion cells by cutting a chunk of onion almost completely through. Then we'll peel back half the chunk and now we should have our thin layer of onion cells. We will add this to a clean sample slide. If the sample slide is dirty, only clean it with a Kim wipe or some other lens-safe material. Now we can add a small amount of water using a dropper or a squeeze bottle. Next, we'll take a cover slip and we'll touch it to the water at about 45 degrees from the slide, and we'll drop it onto our sample. If there are any visible air bubbles, you should probably try dropping the cover slip on again. Next, we're going to stain our sample with either iodine or methylene blue. Iodine has a brownish yellow color to it, and methylene blue is blue. On one side of the cover slip, place a few drops of the stain of your choice, and on the other side of the cover slip, place a paper towel to the water sitting under the slip. The paper towel will absorb the water under the cover slip, and the stain will fill the void of the water that's leaving from under the cover slip. The purpose of a biological stain is to increase the contrast of your image. This allows you to more easily see some of the detail of the structures, such as the nucleus and the cell wall. Make sure you clean up any mess or any material on your slide that could interact with your lenses. Now our sample should be ready to observe under the microscope. So we will make sure the microscope is on low power, and we will put the slide on the stage and secure it with the stage clips. When we look at our sample through the ocular lens or the eyepiece, we have to bring our sample in focus with the coarse focus first, and then we can move the sample around on stage to find a good example of a cell. Now we can increase our magnification if it suits our purposes. Make sure you put your cell in the center of the field of view so it will remain centered when you increase the magnification. As you can see, our onion is clearly made up of many small cells, just like cell theory predicts. In addition, you can see a structure in these cells that we talked about yesterday. What we're looking at right now is the nucleus of the cell. It contains the instructions for the cell to perform all of its functions. It also controls when this cell will multiply to make more cells. As a practice question, I would like you to calculate the estimated size of this onion cell. Pause the video to estimate the size and see if you were correct when you continue the video. When I look at the cell in the picture, I estimate that 3 of these cells could fit side by side in our field of view, or our field diameter. So when we go to estimate our size, we take our field diameter, which is 1,720 micrometers, divide it by our fit number, how many could fit side by side, which is 3, and we get approximately 573 micrometers. That means that this onion cell is approximately 573 micrometers long. As you've seen today, we've learned how to calculate magnification and estimated size using a microscope. You also learned how to prepare a wet mount slide and the purpose of using a biological stain when preparing a sample. In our next lesson, we will start looking at the different structures within our cell and start looking at the function and purpose of each of those structures. Thanks for watching, and I hope you have a great day.